Alright, hello everyone. Today I'm reviewing interphase, which is the first and longest stage of the cell cycle and prepares the cell for division. This is part 1. So what is the cell cycle? So the cell cycle is basically the series of events that a cell goes through to grow and to divide. And to do this, it has 3 main parts: interphase, mitosis, and cytokinesis. So like I said before, grow and divide. Interphase is basically the growth phase. So it's preparing for mitosis, where the cell is going to grow, it's going to carry out its normal metabolic functions, and it's going to replicate its DNA. And then the next piece is mitosis, which is where it's going to actually divide and undergo cell division. We'll touch on that in a later video. And then cytokinesis is when the cells finally split into two and when division is complete. And you also see a little circle over here called G0. This is basically when the cell is outside of its cycle, is doing its normal functions, and it's not preparing to divide and is not going to divide. So now let's break up interphase into 3 main parts: G1, S, and G2. G1 stands for growth 1, S stands for synthesis, and G2 stands for growth phase 2. Now you might be wondering, what are these little red dots on the edge of these phases? So these are called checkpoints. And basically what they are is they are controlled by what is called a cyclin protein. So cyclin proteins drive these by binding to something called a cyclin dependent kinase, which basically interact with the cell via phosphorylation to advance the cellular check. So basically think of these proteins, the cyclin proteins, as kind of like a police force and binding to the cyclin-dependent kinases to detect whether the cell is good enough to continue through the stages and is healthy and is not damaged. Alright, so beginning with the G1 phase, the growth 1 phase. So in the growth 1 phase, the cell is going to check its environment to see whether it should divide or not, and it's going to synthesize proteins for DNA replication. And if it does not pass this checkpoint over here, the G1 checkpoint where it checks for DNA damage and checks for sufficient nutrients, then it's going to go into G0 if it does not satisfy. So if it doesn't have enough nutrients to divide, then it's going to go into G0. Okay, so now let's take a closer look at the G1 phase. So here's a diagram of the cell in the G1 phase. So as you can see here, we have the nucleolus, which is made of proteins and is responsible for RNA transcription and ribosome assembly. The nucleus contains the genetic material, and this little pink things here, these are called centrosomes. The circle here is a centriole, this square is also a centriole. So centrosome is a group of these little centrioles over here. They are responsible for making microtubule spindle fibers, which will become more important in mitosis. And then these lines here, these are very important. So these are called chromatids. The plural is chromatin. Each of them is basically just a long strand of DNA. So there's a red one here, there's a blue one here, this small blue one, a small red one. Each of them are their own long strands of double-stranded DNA. In the G1 phase, they're called uncondensed because they're so long. They're just DNA. Nothing else has happened to them. And now we're going to look at something called the diploid number. So the diploid number basically tells us the full complement of genetic material in the cell. And what that means is basically the amount of unique chromatids in the cell. So there are 4 chromatids which are all unique: red big, red small, blue big, blue small. So the diploid number is going to be equal to 4, and that's represented by 2n. So the sum of G1, the nucleolus is going to synthesize the mRNA proteins for DNA replication, and the cell is going to grow, and it can also duplicate some of its organelles during this phase. So basically, it's just growing and making sure that it's ready for the future phases, such as DNA replication, which occurs in the S phase. So in the S phase is when the DNA is actually physically going to be replicated, and that S checkpoint ensures that the replication is accurate and nothing's going wrong with it. So now let's look at a cell in the S phase. So we can see some things have obviously changed. So first of all, there are 2 centrosomes now. So the centrosomes have been duplicated. The chromatids have also been replicated. So now there's 2 of each variant of chromatid. Well, the diploid number is still 2n because, as we said before, that only represents unique chromatids these this red big, they're not different from each other. They still have the same DNA. And same for all of these. They each have the same DNA as what they were originally. So the diploid number is still going to remain the same. And when a duplicated one and the original, when they're both together, they're called sister chromatids because they're basically twin sisters. And what's holding them together is called cohesion protein, and they basically link the sister chromatids together. They become more vital later on. So basically in the S phase, the centrosomes are duplicated, the DNA is duplicated, and those chromatids turn into two sister chromatids. And then finally in interphase, we have the G2 phase, which is the growth 2 phase, where it's finally going to actually prepare for mitosis by making proteins and replicating their remaining organelles. And the G2 checkpoint verifies that the cell and the DNA are not damaged in any way. So G2 in sum, it's going to look the same as what we saw in S phase, because in S phase, everything's in the process of being duplicated. This G2 diagram is basically like how this cell looks like at the end of interphase. So the cell is going to continue to grow and duplicate organelles and produce mitosis proteins. The summary diagram, we got G1 over here and G2. As you can see, everything's been duplicated because of S. And now we're going to review. So as a review question, what are the 3 phases of interphase and what is the main thing that occurs in each phase? I'll give you guys some time to pause the video and think about your answer, and let me know when you guys are ready to answer. Alright, and we're back. So the answer is that in G1 the cell grows and produces proteins and organelles for DNA replication. In the S phase the replication occurs, and in G2 the cell continues growing and gets proteins for mitosis. So thank you for watching my video on interphase, and see you in the next one.